Now, students, when we observe the chromosome into the metaphase, now there is one technique in which the cells are broken up and their chromosomes are counted. And this particular technique, they restrict the chromosome into the metaphase. So whatever chromosome spread out on a slide, that is called as the karyotype. Now this karyotype is usually is used to study the chromosomes, such like chromosomal abnormalities or cytogenetics, all this. But see, karyotype is just spread out of chromosome, understood on a slide. But if these chromosomes are properly arranged in the decreasing order of their size, this is called as the ideogram. Okay, we need to remember there is a difference. Karyotype, usually students get confused that the total chromosome of the any species is called as what? It is called as the karyotype. Okay, and proper arrangement of the chromosome in its decreasing order is your ideogram. Now, on the basis of character, the chromosomes are classified as the autosome and the sex chromosome, or you can say the heterosome. So this is called as the classification of the chromosome on the basis of its character. So when we talk about the autosomes, so autosomes, they just transmit the somatic characters only, like generally we can see they carry the genes of height, body structure, length of appendages, color of skin, our structure, like shape of eye, ear, nose, color of eye. So all such things are present on the autosome. But when we count the number of the autosome, so that particular thing is more. Autosome numbers are always more in the case of any any species, chahe wo human ray or any other type of, whether it is birds or any type of animal species or any type of sexually reproducing species, their number of autosome is always higher than the number of heterosome or the sex chromosome. So in the case of the human, we have the 44 chromosomes, which represents as autosome, or you can say the 22 pair, and only one pair represents the sex chromosome. When we talk about the sex chromosome, so these are the chromosomes, they are responsible for the determination of the sex of individual and inheritance of the sex linked character. Now we have learned about the X linked inheritance, right? So whatever inheritance are observed, that is observed on the sex chromosome. Numerically, when we count, so again this is only one pair. In case of human, only two are there. Now when we talk about human sex chromosome are designated as X and Y, right? So we all are going to learn about the inheritance of the sex chromosome in earlier. Other, when we talk about the male, so male is having one Y chromosome. Now see, this X chromosome, it is present in both males as well as the female. When we see its structure, so it is bit straight and it is longer than the Y chromosome. When we talk about the length, so its length is 5.0 to 5.5 μm, that is micrometer. And let me revise, 1 micrometer is equals to 10 to minus 6 meters. So can you imagine, so tiny it is. Now if we will talk about its type, so just now we have learned that it is metacentric in nature. It is larger, so that's why it contains large amount of gene. Now see children, we have learned in earlier lecture about the euchromatin and heterochromatin, you remember? So euchromatin is the region where more and more active genes are present. So this X chromosome, it is having more, or you can say the large amount of euchromatin and a small amount of the heterochromatin. When we talk about the non homologous part of X chromosome. Homologous part matlab, now see, these two chromosomes are homologous, having the same length. But if we will talk about the non-homologous part, that means this region from here till here, this is the non-homologous part. That means it do not pair. Now remaining pairing will occur. Actually centromere to centromere pairing will occur. So this region which do not matches, let me take the another diagram. But in general, you remember the part which does not match. Here centromere to centromere matching, or you can say they will lie up, and thereafter whatever part which is not matching is called as the non-homologous part. So this non-homologous part that will share, or that will be the part where the X-linked genes are present. That's why this particular region makes the male as hemizygous. We all remember when we were talking about the pedigree. So that particular time I told you that the males are hemizygous because of absence of one X chromosome. Now why this Y chromosome is not compensating? This Y chromosome is not compensating. Just a minute, let me take another diagram. I will show you another diagram for non-homologous. Just a minute. This diagram I immediately clicked from your textbook. So see, this is the non-homologous. When centromere to centromere will join, so this part is the non-homologous part. That means this is not matching. When the chromosomes, you know that this homologous chromosome, they pair at the time of the division. So that particular time, this non-homologous part will not compensate, and all the X-linked genes in male, due to the absence of this non-homologous part, counterpart on the Y chromosome, causes the X-linked disease more and more in the case of the males. Understood? So this is the reason. So what are the common traits or disease related to X chromosome? Yes, X-linked recessive disease, which are more and more which have more and more chances in the case of the human. Can anyone tell me what are the X-linked recessive disease in which the males can be affected more and more fast? We had discussed about hemophilia and color blindness, right? These are the X-linked trait which have chances to be present more and more in the case of the males. That means male are more susceptible for those diseases. Now when we talk about the Y chromosome, so Y chromosome is present only and only in males, and when you will observe, it is shorter than the X chromosome. Now this particular chromosome length is only 2 millimeters. See, this is the reason. When we talk about the X chromosome, it is 5 millimeter and it is only 2 millimeter. That's why it is not matching properly into the height, and that's why the extra region that counterparts the X-linked genes oid. Now when we talk about the type, so it is acrocentric in nature. It contains only small amount of euchromatin. That means it may have only little part of the functionally transcripting genes. Understood? And whatever genes are present on the non-homologous part of the Y chromosome, now you will be thinking, ma'am, how the Y chromosome is having the non-homologous part? Because it is slightly tilted. Okay, so that's why it does not matches here. So for that particular reason, these genes are this many only, and these are called as the Y-linked genes, and the inheritance which is present on that is type of inheritance is called as the Y-linked inheritance, and that particular region is called as the Hollandric region. and these genes will transmit only linear way, that means from father to the son, and all the offspring will be affected. So this holendric genes example, disease related or the trait related to this holendric gene example is the hypertrichosis. So this is responsible for the linear inheritance, that means from father all the sons will be affected, and same way proteidry we can say this disease will be limited or present only and only in the males, understood, like ears on pinna and all such genes are holendric genes. So these particular traits that will be observed into the males only. So this is all about the classification of the chromosomes, and next lecture we all will be talking about the another phenomena that is called as the endomitosis. So this is all about this lecture. Any doubts, students? Yes, do you have any doubt?
No, ma'am.
So we'll stop at this note.